Describe the first fertilization changes in the life history in Polycythemia. In Polycythemia, first fertilizations involves the transformations of the corpus genomes and supporting cells into a cystocarp, a nutritive structure that develops into a dipole corpus prophytes, uh, completing the triphasic life cycles. Key steps include the corpus genomes, filament elongating, filament elongating, filament elongating, and axillary cells forming, completing to the corpus genial, completing to the corpus genial, the dipole zygote nucleus migrating to axillary cells, and the eventually developments of zoonoplast filaments. Corpus spongium within the protective pericarp. Here a detailed breakdown. Here a detailed breakdowns. First fertilization changes. Corpus genial filament developments. Uh, Corpus genial after the fusion of male and female nuclei. The two cells lateral sterile filament. Lateral sterile filament. Lateral sterile filaments near the carpogenian elongates and divides to form 4 से लेकर 10 cells nutritive filaments and with it base also forming two cells filaments ठीक ना axillary cells diploid axillary cell below the carpogenum buds of a haploid axillary cells which forms a tubular connections with the carpogenal base. इसके बाद फिर तीसरा में क्या से हलै तीसरा में अथी हलै zygote migrations. The dipole zygote nucleus one nucleus travels through the two axillary cells while the other stage in a carpogenum which eventually degenerates. Blast filaments. The dipole nucleus axillary cell divides, initiating the developments of zonoblast. Initiating the developments of zonoblast, which grow upgrades from the axillary cells. इसके बाद cystocarp formations. The zonoblast filaments. The zonoblast filaments produce dipole corpus spongia, which from non-motile corpus spores. The entire structures, including the sterile filaments and the dipole corpus spongia, dipole corpus spongia, ठीक ना dipole corpus spongia becomes an enclosed by sterile filaments and forming the protective pericarps. Cystocarps create the cystocarp. इसके बाद फिर क्या बोलते हैं carpus spores release and germinations. Dipole carpus spores are released and germinate to form a dipole carpus spores, and which forms a haploid carpus spores, carpus spores, and completing the life cycles forming by male and female gametophytes. This entire sequence establishing the parasitic the dipole tetrasprophyte phase. The life cycles of Polycythemia, characteristic features of red algae. Answer complete.